you for. Yeah, we're very, very happy to have you all here and have Diego here. So Diego got his undergrad at the very in Cuernavaca, and then he went ahead and got his PhD at UCLA with John November and then Chris there as well. And he stayed in California to do a postdoc with Ron Slatkin full time. He's been doing very exciting theoretical and then theoretical to applied work in population genetics. Now he's got returned to Ciencias en Loma, but now in Juriquilla, it's on campus there, where he's a faculty member, and I'm very pleased to come and tell us a little bit about it today. So let's welcome Diego.
Excellent. So thanks a lot for that kind introduction, Rory, and thanks a lot for inviting me here to have a chance to talk to you a little bit about the work that we're doing in the lab. So mostly I'm gonna tell you a little bit about some of the work that we have been doing trying to study how natural selection acts. So let's see. Okay. All right. So natural selection is an evolutionary process that exerts its strength on phenotypes, and this has impact on both patterns of genetic variation and also patterns of phenotypic variation. And in terms of genetic variation, the two, I would say two of the ways that genetic variation can be impacted is that, for example, you can have alleles that are advantageous that will rise in frequency above time. And there are also deleterious alleles that with decrease time. And the most common type of genetic variation that is acting, as we know, for example, in new non-synovial mutations, is that most of the new mutations tend to be deleterious instead of advantageous, and that's what we currently know. And it also, natural selection can act on patterns of phenotypic variation. And here I'm showing you one particular plot where on the x-axis we have the value of a particular phenotype, and on the y-axis we have survival, which is a proxy for fitness. And what you see is that in this particular plot, as you move farther away from a particular value, then the fitness can decrease as you move farther away. So this is a type of selection that is called stabilizing selection. And new research that has been going on in humans has found that many of the traits that are that people analyze tend to be under some sort of stabilizing selection. So these are two ways that natural selection can change patterns of genetic and phenotypic variation. And the central focus of our group is to study how natural selection is acting across the genome, and then how does natural selection modify patterns of space-time patterns of genetic and phenotypic variation around the world. And so today I'm going to tell you a little bit about a few of the projects that we are currently doing in our lab, and I'm going to tell you like the basic ideas behind them. And first I'm going to tell you about one project that we're currently doing, which is trying to infer something called the distribution of fitness effects using genealogical trees. So this is a project that has been led by a graduate student in our group called Tananisa Raras, with collaborations from Christian Huber and Charleston Chiang from Penn State and USC. So, so this is a super important distribution of probability in biology called the distribution of fitness effects. So the distribution of fitness effects is a central probability distribution in evolutionary biology. And that particular probability distribution, it tells you if you have a new mutation that appears on the genome, then what's the probability that that particular mutation is advantageous, or what is the probability that that new mutation is neutral, or what's the probability that that mutation is deleterious, and also how deleterious is going to be that particular mutation. Usually the way we parameterize this particular this distribution is we use something called the selective coefficient, where, for example, if you have one particular fitness, then if you have a heterozygous copy, then, and if you assume that this is additive, then maybe the fitness for this particular genotype is going to be 1 plus S. And if you have a homozygous genotype for this particular allele, then maybe your fitness is equal to 1 plus 2S. And then this particular selective coefficient can be negative, which means that the mutation is more deleterious and it decreases the fitness of the individual carrying that particular mutation, or it can be maybe beneficial. And for example, in non-synonymous mutation, what we know is that advantageous mutations tend to be rare, and most of the mutations tend to be either neutral or slightly deleterious, and then you have a fraction of mutations that are strongly deleterious. So this is actually a DFE that has been inferred in humans, and this is the shape of the distribution. So we are particularly interested in characterizing distribution very well. And we are interested in it because of many factors. And one of those factors is that actually characterizing this distribution of fitness effects can be key for conservation efforts. So right now in conservation genomics, there's a new paradigm that has been emerging where you can collect genomic data. And then based on that genomic data, you can infer tons of parameters about one particular species that you're studying. You can demographic parameters, selection parameters, or mutational parameters. And then you can do simulations. Just like ecology and life history data. And then you can, based on those simulations, you can compute the extinction risk of a particular species or organisms, and then you can try to make management decisions. So inferring accurately evolutionary parameters is key in these efforts, and accurate inference of those parameters is vital. And one of those parameters that you must set up in, if you want to try to follow this particular paradigm, is you need to characterize very well the distribution of fitness effects so that you know what proportion of mutations are going to be deleterious or neutral or advantageous, because that's going to guide. If you do that very accurately, then you can get a better sense of what's the chance of that particular population to become extinct or not. So that is key, right? So there are many methods that have developed to estimate that particular distribution of fitness effects, and they use different types of data. And one uses one particular type of summary statistic called the site frequency spectrum, which basically summarizes the allele frequencies over many different sites in the genome. And there are also methods that use multiple summary statistics or that use haplotype data. So there are many methods to infer the distribution of fitness effects at the moment. And so the thing is that many times when we work where we want to make assessments about the extinction risk, many times you have small sample sizes. So when I talk about small sample sizes, is that less than 50 genomes, and you want to make a management decision. So one particular caveat is that inferences based on commonly used summary statistics provide less accurate inferences with low sample sizes using methods such as FitTally, and this is something that has been known before. So the challenge here was to see if we can develop an inference method that can infer more accurately the distribution of fitness effects when you have maybe low sample sizes, and overall, if you can get better inferences of the distribution of fitness effects. So then something that has been happening very recently in evolutionary biology that has been very exciting is the chance of analyzing whole genome data using something called the ancestral recombination graph. So the ancestral recombination graph basically tells you what are the relationships between a set of samples that you get across many species, across all of the genome. So one way of summarizing this is that, for example, you can look at the genome, and in the genome, in every section of the genome, you have one particular genealogy that tells you the relationship between all of the samples that you have taken. And then you can look at this across all of the genome. And something that is particularly exciting is that all of the evolutionary information that you can get from that sense of sequences is encoded in the ancestral recombination graph. So if you are able to develop a method or a model that can asks about some particular evolutionary process using the ancestral recombination graph, in theory, you can use all of the data more efficiently to try to estimate how some evolutionary process acts. And it should be more efficient than using some summary statistic. There's a ton of information there that we can use to do evolutionary inferences. So that's why we wanted to try to do inferences using the ancestral recombination graph. So broadly, what I wanted to say is that leveraging genealogical something that is beneficial for evolutionary inferences with low sample sizes, and that's something that we have been observing for some time so far. So for example, there's a particular method called that has a genealogical thinking in mind, where the idea is that you can sample a couple of chromosomes, and then you can look at the time to the most recent and common ancestor in those chromosomes across all of the genome. And then that time to the more recent common ancestor is going to get the numbers. And so you can use that information about the time to the more recent common ancestors between pairs of chromosomes. And you can also principle that small population sizes give you shorter coalescence times, whereas big population sizes give you bigger coalescence times. And then in the top plot, what I'm showing over there is that the distribution of heterozygous sites across the genome tells you something about the distribution of times to the more recent common ancestor between pairs of genomes. And basically, you can code a model that, based on that distribution of times to the more recent common ancestors, you can infer an effective population's time you can fit changes in the effective population size over time. So in essence, you can get very good inferences of how population size changes over time. And this gives the idea that if you actually have a genealogical thinking in mind, you can, get a ton of information from a data set that you have. And for example, here they do that with a pair of chromosomes. So that's more motivation. You can ask the right questions using a genealogical thinking in mind. You can get a ton of information about past evolutionary processes. So based on this idea, we decided to try to estimate the impact of natural selection using genealogical trees. So there has been previous work. I'm showing 2 papers over there that a framework that we're trying to exploit to look at the distribution of fitness effects. And when they did was they said, well, we have this part that appeared here. And then over time, we can look at the number of particular mutation. How many lineages have that particular inferences of how selection is acting? So previous work has done that for advantageous mutations, but we thought, well, what we use that source of information to infer a distribution of fitness effect. So how will we do that? Well, the way we will do this is look over different slices of time. So for example, at T1, we're going to see how many lineages contain that derived mutation. And then at time T2, we're going to look at how lineages contain that derived mutation. And the same at T3 and the same at T4. So 2 lineages have the mutation, and then here no lineage has the mutation. So we're going to use this particular information to try to infer how selection is acting, because we can actually process from a ton of trees that we have over the genome, and then we just look at how many lineages have this particular mutation. So then we went on to set up a likelihood framework where the idea is that, for example, you have a tree, and for that particular tree, you ask, what's the chance that I have this number of lineages with the derived mutation, given some value of selection, and given that I'm looking at some particular time. So you can look at this you can fill up a table, where in this particular table, you look at how many lineages have that derived value for time, and you can do that for different times. And basically, this probability is something that you can compute via Monte Carlo simulation. That's the way we do it. And then we can set up a likelihood equation where what we look is, what's the chance of having a certain number of lineages derived allele, given some value of selection, given that looking at a particular time, and then I look at different time slices. And that's the way we're going to do it. And we can repeat that exercise with a ton of genealogies. So you can leverage information from a ton of genealogies and then compute what's the likelihood of having some value of selection, given all of these genealogies and this information of the number of lineages that have the derived allele over time. So we went on and we did this. And so basically the way we do it is we compute the likelihood value for different values of selection, and then we just try to find out where we maximize our likelihood function. So we went on and we tried this, and here are some results simulations. The real value of the parameter is a function of the population size multiplied by the selection coefficient, which is the selection coefficient I was telling you before. And here we look at results where we have a value of selection equal to 0, 0.01, 0.1, 1, 10, 100, and 1,000. And what we saw that when the value of selection was bigger than, when n times s was bigger than 1, we get accurate inferences of selection. But then as the selection value gets lower, it's hard to differentiate between neutral from neutral mutations. And these simulations were done for a population size of 10,000. And we saw that for these particular simulations, the inferences are accurate up to that particular selection coefficient value. But in essence, like the value of n times s gives the cutoff for when estimations of selections are going to work well, and then when it's going to be harder to estimate selection. So that's where we're currently at. And then over in the past slide, I showed you how we can do that for one particular point selection coefficient, but you can also do the same to infer parameters of a distribution of fitness effects. So in humans, what people have seen is that the gamma distribution provides a good fit many times to the distribution of fitness effects. So people have seen that based on that observation, you can try to set up a gamma distribution or any other probability distribution that you wish. And then you can try to infer the parameters that define that distribution of fitness effects. And we have a likelihood scheme where the idea is that, so this particular term, we got it once again using Monte Carlo simulations. And this one is based on the probability distribution that we want to infer. But basically, we can ask, what's the probability of having this point selection value of selection given the parameters of the distribution of fitness effects, and we can do an integration. And in essence, we can get a likelihood function for the value of the parameters. And then we can try to infer what parameters best describe this distribution. So we went on and did this, and here I'm going to show you some results. So this is the a distribution of fitness effects as what's inferred by in humans, in Boyko, and these are the parameters of the distribution. And we did some simulations. And in those simulations, we tried our method, and we saw how the two parameters of the gamma distribution of fitness effects infer the real value. And we saw that, overall seem kind of close to the actual value of the distribution of fitness effects. And exactly what in terms of how distribution of fitness effects looks, based on these parameters that we infer, this is the actual probability mass of the distribution of fitness effects. And this is shown inside of every probability of this distribution. And then this is what the median value is shown over here over 50 simulations. And this is a 5 and 95% confidence intervals that we got from our estimates. And what we see is that broadly, we kind of get the probability mass that you expect on every one of these selection bins. So we see that our method gives, reasonable estimates of the distribution of it. Yeah, Karen, Andy. Yes, exactly. Yes. And the way we set up these simulations, these particular simulations assume MB mutation is independent, and the selection coefficient is drawn from a distribution of fitness effects. Make sense? Oh, yeah, please. That's a good question. So we assume that for every mutation, for every side of the genome, there's a mutation, and that mutation has the same selective coefficient across all the history. Okay, so what we are also trying to look at the moment is to see how these inferences work with low sample sizes, and that's something that we're currently tackling. And we are trying from 50 genomes to 10 genomes to 2 genomes, and we see that it sort of works kind of in the same way, where you get I think that was missing here, but for nearly intermutations, it's harder, just as in our previous simulations where I used 100 genomes. So we kind of get the same results. So this is encouraging because it's we're doing some tests right now, more tests right now, but it means that maybe we can use this method to do inferences of the DFE with a lower sample sizes, and that's something we are currently investigating. So that's where we are currently working on. So in essence, we have developed a method to infer the distribution of fitness effects by leveraging combination graph. So far, it seems like our method can be applied in scenarios where we have low sample sizes, and we're going to apply this methodology to the extinction risk and also infer the impact of selection on new mutations of the particular function. So that's our things. And we are also doing some more tests to see how well we can infer the distribution of fitness effects using also data from simulated data from other simulators, such as SLIM from standard, and also from standard PopSim. So that's where we are. And okay, so I want to tell you some other projects that are going on in the lab. So we're looking the impact of natural selection on the prediction of complex traits using ancient DNA. And this is actually work that has been carried out by Valeria Ñorbe, who is an undergrad student also at UNAM, in collaboration with Machal Sohail at UNAM and Emilia Huerta Sanchez at Brown. And actually, we have a preprint explaining some of this work that you can be interested. And so an area that is of great interest lately is to infer phenotypes based on genetic data. So people have been trying to do this using data from individuals, and the idea is you have genetic data, and then based on that genetic data, you can try to predict phenotypes such as human height, for example. And here, for example, people have used genetic data to try to predict the femur length, and then they try to do that, for example, for male and females, and try to see how well using genetic data you can predict a trait. Basically, this is done something called polygenic risk scores or polygenic scores, which predicts traits based on genomic data and estimates of the impact of variants on a trait. So there are many variants across the genome that can impact a trait, and you can get an estimate of how it impacts the trait, if it increases or decreases the value of the trait. And most of these things are done, basically, people use present-day genomic data to estimate the impact of a variant on a trait, and then they use that estimate of the effect of a variant on a trait to try to predict ancient phenotypes, in essence, phenotypes from ancient individuals. So, but one thing that has happened is that when people have tried to estimate phenotypes from ancient individuals, the prediction does not work very well. So, for example, for length, they have seen that polygenic risk scores predict like 6.3% of the variance in femur length, which is very small. And it's particularly striking because variation in height or femur length, which is a proxy for height, is highly dependent on genetic factors. People have seen that 30 to 90% of the variation is explained due to genetic factors, but even if there's a high genetic contribution, the predictions do not work very well. And there's a question of why is this happening? There are many things involved. And one of the main questions people have been trying to tackle is what evolutionary factors could be contributing to poor predictions of phenotypes via polygenic scores. So one particular force that has very recently been recognized to be very important acting on humans, which is stabilizing selection. So stabilizing selection is a force that is acting on many phenotypes of humans. And once again, here, stabilizing selection is a force where you have a phenotype value, fitness, you have it here on the y-axis. And then as you move farther away from a particular phenotypic value, then fitness is going to decrease. Here, survival is a proxy for fitness. In humans, for example, one study found that 26 out of 70 study traits are under stabilizing selection. And another study found out that 21 out of 27 study traits are under stabilizing selection. So it seems like this is a very important force acting on many human traits. So we decided to figure out if actually this particular evolutionary force could be problematic to, could be a source of confounding to traits in ancient humans. So the way we try to tackle this is using a tool called SLIM, which does simulations. So we try to do simulation. So we did simulations with 20 dependent region of 25 kb. And the idea is that in these simulations, we try to, we simulate genetic variation that is acting on a particular trait. And then the idea is that we are going to assume that we can predict the accurate a trait, we can accurately predict the impacts of any genetic variant where we have variation in the present, and we use that information to try to predict the phenotypic value of an ancient individual. So we are going to try to do that. And so this is the lesson that I wanted to, these are some results that we did when we tried or doing our simulations. So here, simulations were, so this is the present, and this is 100, 200, 300, and 400 generations before the present. And we use this particular time span because that's the time span of 0 to 10,000 years ago, which is where you have most of the human genomic data and where people have been trying to predict phenotypes. And we did simulations where in these simulations, we take all the variants with a frequency bigger than 0.5% in the present, assume that we know their genetic effect, and we try to predict ancient phenotypes in the past. And when we do this, and here I did simulations with two values of heritability, which tells you how much of the variance is explained by genetic factors. And when all of the variance is explained by genetic factors, we see that we can, over here, I have an R-square where I compare the phenotypes that we have in the past compared to what we've predicted using polygenic scores. And what I see is that when the heritability is equal to 1, basically we can predict very well the trait under neutrality. And when the heritability is equal to 0.5, which means only 50% of the variation is explained by genetic factors, basically the R-square value between the predicted phenotype and the real phenotype is close to 50, which is what we have expected under neutrality. Okay, but that's what happens. When we have stabilizing selection, then the prediction is not as accurate. So over here, the heritability value is equal to 1, which means all of the trait is explained due to genetic factors. All the variation in the trait is due to that. And then over here, so stabilizing selection can be thought of as a Gaussian function. And in this particular Gaussian function, we have a parameter called W, which tells you how strong is natural selection acting on the trait. And basically, when W is lower, then there's a higher strength of stabilizing selection. So in this particular scenario, when W is equal to 1, it's stronger than equal to 5. And when it's equal to 1, it means that that Gaussian function is more steep. And when it has a higher value, it means that it's not as steep in essence. But what I wanted to say is that when stabilizing selection is stronger, then in times to close to 10,000 years ago, very poor predictions of traits, just due to this particular evolutionary process. So stabilizing selection has an important impact on the prediction of traits. So that's something that we think that that's something that should be considered when people try to infer complex traits in ancient humans, because that is a factor that can affect the prediction of traits. So that's one thing. And we also try to explore other evolutionary process called directional selection, where we have a phenotype value, which changes from a value to 0 to a value of 1. And then basically, in the past, the optimal value is 0, and then it's equal to 1. And in this particular scenario, we also saw that depending on what is the variance of the effect of the traits, basically, there are new mutations that are appearing, and if the new mutation have a stronger effect on the trait, then here sigma tells you what's the variance of the impact of a new mutation on a trait. And if it's bigger, then it means that there's a bigger contribution of a new genetic variant to the trait. And if that value is bigger, then the predictions tend to be lower, which means that if every new genetic variant that appears has a stronger effect on the trait, then under directional selection, then also your predictions are going to be worse. So this is so directional selection is another part acting on some phenotypic traits. As far as we know, case for directional selection acting on a trait, but we don't know if directional selection is acting on more traits, but if it is acting, then depending on the impact of new mutations on the trait, there can be impacts on how well natural selection is predicting, how well we can predict traits in the past using ancient genetic data. And what does this happen? Well, it happens because of, I'm going to give the long summary of why this happens, but there are different patterns of shared variation between ancient and present day population when there are different evolutionary processes acting on the trait. So under neutral evolution, if you assume that the trait is neutral, then whether a particular loci is conserved or not is not going to depend on the effect size of a new mutation. However, under stabilizing selection, the mutations that have a higher impact on the trait tend to be deleted from the population, and that affects your prediction of ancient traits. And that is something that we see over here. Effect where traits that move you closer to the new optimum tend to be conserved, and lost alleles that move you farther away from the optimum tend to be deleted. And basically, those three patterns of those particular patterns of shared genetic variation, which here I'm summarizing, are those that are affecting the prediction of complex traits. So in essence, so stabilizing selection decreases the accuracy of phenotypic predictions via polygenic scores. Stabilizing selection promotes the loss of high effect alleles. And directional selection also decreases the accuracy of phenotypic prediction with polygenic scores. And directional selection promotes the loss of alleles that move individuals farther from the optimum phenotypes. So basically, the impact of natural selection acting on traits should be, we believe it should be taken into account for predicting traits in the past. And we think that maybe doing some simulations where you know how strong is stabilizing selection acting on a trait should be conducted so that you can get an idea of how well you can predict traits in the past. Clear? And this is a project that actually, and the third project that is also still on the making is doing space-time inference. And this project that is being carried out by Alessandro Lopez, which is in our lab. And we know that natural selection drives the geographical distribution of alleles. Oh, we know that, for example, when you look at in humans, depending on which region of the world you're looking at, you can see different variants that are under positive selection and are advantageous. So there's a geographical component to which genetic variants are going to be selected in which part of the world. So because there's an important geographical component, then we talk to get a look at this problem and try to see if we can use geographical information to try to infer how natural selection is acting. And there are some previous work that has been done on this particular space. So for example, Mukto-Pavelam developed a method to use ancient DNA, a present day DNA, to infer the impact of natural selection acting on a new advantageous mutation. So there has been some work done So, but we were interested in this particular line of work because some of the methods that have been developed do not consider demographic events such as large distance migration. So the papers that we have already been conducted do not putatively deleterious alleles, and basically the deleterious alleles will decrease the frequency of an allele in a particular region, and basically the deleterious alleles tend to be not as much expanded as neutral or advantageous alleles, so we wanted to take a look at that. And most of the studies have been focused on genetic data from Europe, so we wanted to see if we can actually use some of the new results that have been obtained to estimate, to look at selection in a broader genetic sample. So we have been trying to develop methods to estimate migration, dispersion, and demographic events and selection. The idea is that we're going to try to use allele frequencies, and we're going to use temporal information and geographic application to try to estimate many of these factors. And the idea is that at the moment, we try to put the location of those populations in a particular matrix, and then we're going to try to estimate the allele frequencies of those alleles over that particular grid in a particular generation. And overall, the idea is we're going to develop a likelihood function where we're going to try to estimate parameters that are related to demographic processes and selection in a particular likelihood scheme. And I'm going to tell you a little bit more about this. So we are currently working on looking at this using a model where, in this particular model, this is constructed based on previous work by Pavela, Novembre, Fisher, and Halasek and Fisher. And the idea is that first on the diffusion equation coefficient, which tells you how much you can move on the X and the Y axis. So that tells you how much you're going to move in a particular geographic. And then we have another parameter back on natural selection. I mean, it could vary in space and time theoretically. We haven't looked at that, but the idea is we're going to look at deleterious and variants that decrease in frequency and advantageous variants that increase in frequency. We have an stochastic term to model changes in population sizes and drift, which is the first term. And then we were thinking about adding another term to model long distance migration, and we're still working on those. Actually, those two last terms, we're still working on incorporating them. But that's so the idea is to use that particular equation. And our idea is to first infer migration demographic parameters via likelihood function. So the idea is first we're going to estimate demographic parameters in putatively neutral alleles, and then we're going to estimate natural selection acting on putatively selected alleles. And the broad idea is that maybe we cannot estimate selection acting on a putatively, for example, deleterious alleles, because it's very hard. But if we take a ton of deleterious alleles, maybe we can estimate the action of selection acting on that particular set of deleterious alleles. So this is what we're currently trying to work with. And we are using, once again, SLIM to do simulation. So far, we have been working on stepping stone models where we have a grid of 3 by 3 or a 5 by 5 or 10 by 10. Doing some simulation model would work on this particular setting. And so first, some of the results that Sandro has seen is that, so he has done composite likelihood estimates from 50 estimates of the dispersion coefficient. And basically what he has seen is that based on this likelihood function that he has, he can estimate, he can basically estimate well how much an allele migrates between neighboring populations. So the real value over there is the red bar the red line over there, and the estimates are shown over there in the y-axis. And basically, with 25 DIMs or 100 DIMs, we see results of the diffusion of those diffusion values or migration values. And then we also got estimates when we decrease the value of migration between neighboring DIMs. And we also saw that not so unreasonable estimates of the diffusion of those particular alleles. So that's what we're currently working on. And we're also trying to use this model to estimate selection coefficients. And over there, the real value of the selection coefficient is shown with the red line. When the number of DIMs is high, our method seems to be working better than when the number of DIMs is small. And when the number of DIMs is small, it's harder to estimate the selection coefficient well. So we're working with this still, but we're trying to develop a method to do inference of demographic parameters and selection coefficient values. And I mean, it seems like our model is able to estimate seems to be able to estimate selection and diffusion coefficient. The migration rates and diffusion coefficient estimated and concordant we have seen, doing some tests, we have seen that our model is limited in its ability to infer small diffusion values. And basically in our simulations, we see that our estimates tend to improve when we simulate a bigger number of populations. And here we're only using discrete populations, and we want to move to doing simulations in a continuous space. And that's something that is still in the works. They're learning how to do best. So that's where we currently are. And yeah, this is brought to you today. It's a sample of some of the ongoing work in our lab, and I want to thank you for your attention. Thanks. Oh yeah, please. Yeah.
Effectively calculating summary statistic from the surgery and matching that to patient. And wondering if the inference part of that, like, are you somehow incorporating that additional noise from the inference process into the simulation?
Yeah.
So how do you go about that?
That's a good question. And that's something that is on the back of our minds of how to do better. And the way we are currently testing it is we're currently doing simulation using a standard PopSim. So in a standard PopSim, you can simulate like a whole chromosome. And then in that chromosome, you have some genes over there where there are non-synchronous mutations where you want to estimate the impact of selection. And what we're currently doing is we do simulations on a standard PopSim, and we take the tree, and we estimate the DFE based on the actual tree. And then what we do is we feed that chromosome into the software called Relate, that does arc inference, and then we do the estimate of selection based on that. And so far, the results from Alan have seen that, the estimates pretty similar based on that. So that's the way we incorporate that noise at the moment. Yeah. Exactly. So at least sufficiently robust for inferring this thing. Yeah. Oh. Mm-hmm. Yeah.
So the simulation goal essentially is
That is something that I have not talked about. That actually sounds like a great idea. So I would be interested, I would be interested in seeing if you actually use, the decreasing accuracy over time to estimate stabilizing selection and seeing if those estimates are concordant with other methods to estimate stabilizing selection. That sounds great. Yeah, I don't know. I haven't thought about that, but I think it could work. Yeah. That's, yeah, that's a good question. So people, other things that could be affecting it could be changes in the environment over time. So for example, her height, the environment is very different to what is now to maybe what it was 10,000 years ago. And maybe that's prediction of traits. So that is one particular thing. And another thing that could be affecting is, well, broadly the environment. So for example, we can think about nutrition, can be one factor, but also maybe some, people have wondered if maybe some particular thing that caused a disease in the past could also be affecting height. So broadly, environmental factors could, in the present or in the past that are different, could affect the prediction of the trait. So that's another fact. And I think that needs to be looked at. Yeah, but yeah, I think that needs to be looked at. But we haven't looked, but yeah. Thanks. Thank you.